Hi, good day everyone. Right now, we will present about plant-based galantamine for galantamine-based drugs. In this video, we will cover the introduction, the process of making galantamine-based drugs, and the conclusion. Alzheimer's disease is a type of brain disorder that can affect patients' memory. This disease is ranked 6th in cause of death with 37.3 deaths in 100,000 people. Because of this, drugs that can be used to treat the disorder are needed to reduce the number of Alzheimer's patients. It has been shown in several papers that galantamine can be used to treat mild and moderate Alzheimer's patients. Galantamine itself is a naturally occurring alkaloid that can act as an acetylcholinesterase inhibitor. Galantamine-based drugs have been sold in the form of tablet with Rasadin as the brand. These drugs can delay the emergence of Alzheimer's disease symptoms. This is an overview of the entire process that consists of galantamine source selection and bioreactor selection as the upstream processes, continued with galantamine extraction, purification, filtration, and formation of the tablets as the downstream processes. There are several options of plant for the source of galantamine as it's shown in the video. However, the best option is Leucojum aestivum due to the large size of its bulb.
For the bioreactor that is used, we have selected bubble column bioreactor compared to Rita-T's bioreactor, due to bubble column bioreactor can be modified to fit the culture, and also parameters can be adjusted. Therefore, a better yield can be achieved by selecting bubble column bioreactor.
In terms of the extraction process, we reviewed two setups that can be used, the Soxhlet and ultrasound extraction. The Soxhlet extraction system is a lab-scale conventional technique. However, the ultrasound extraction system is a great alternative as it solves the issue of time, solvent, and power-consuming techniques. SPE purification employs the chromatographic technique that uses a stationary phase and mobile phase to purify the sample, while fresh chromatography is a type of column chromatography that uses positive air pressure to push the solvent down. Since SPE is the commonly used technique to purify the galantamine extract, it is the chosen technique for our review paper.
Filtration is usually done after the purification and extraction process, as it is necessary to ensure complete purification of galantamine as it will be used for medicinal purposes. Therefore, to avoid the presence of compound traces, filtration is usually conducted. Filtration method will vary depending on what process is done. Methods such as evaporation, sieve, and vacuum are examples of filtration method that is used.
There are two methods that can be used to convert galantamine to galantamine hydrobromide. The first one is using solid filters, which can generate 80% yield of galantamine hydrobromide, and the other one is using hydrobromic acid, which can generate 85% yield. This is why hydrobromic acid is more preferable compared to solid filters. The mixing process to produce galantamine tablet can be called granulation process. The most common method for granulation process is direct compression and granulation. Compared to granulation, direct compression is less favorable. Even though direct compression is simpler, faster, and cost-effective, this method has low flowability and low uniformity. For the granulation itself, there are two different methods, wet and dry granulation. As the name suggests, wet granulation involves liquid, whereas dry granulation is done without any liquid. Between these two different types of granulation, wet granulation is the preferable one with higher uniformity and less dust generated. In conclusion, every single step and method chosen can affect the attempt to obtain high quality and high quantity of galantamine-based drugs. Based on the literature review, Leucojum aestivum can give the highest galantamine yield compared to other plants. Bubble column bioreactor is the more suitable one compared to Rita bioreactor. Ultrasound extraction is more preferable for extraction method, while solid phase extraction is more preferable for purification method. Using hydrobromic acid can give higher yield for galantamine hydrobromide. Lastly, the preferable granulation method is wet granulation. That is all from us.